Hello and welcome to Mr. Saucedo's YouTube videos. Today we're going to be talking about the stages of mitosis. So there are four main stages in mitosis. We're going to go through the details of them. Keep in mind that there are in-between stages in mitosis. These are the four main ones. So the other in-between little bits we don't need to know about. We just need to know the four main phases. So the first phase is called prophase. Okay, we're going to label some important little bits. This is our nucleolus right here. Remember, it manufactures ribosomes. These little guys right in the middle of these chromosomes, these are our centromeres. Centrioles, remember, only found in animal cells. They help divide. Chromosomes, that's that X-shaped thing. This little dashed or dotted line is supposed to represent the nuclear envelope. And then these lines are called spindle fibers, and we'll talk about them in a second. So what happens in prophase? It is the first phase of mitosis. Remember, we've gone through G1, we've gone through S, we've gone through G2, and now we're starting mitosis. Okay, so that's why we have chromosomes and not just, just some sort of weird chromatin mixture. So first things first, the nuclear envelope is going to dissolve, exposing all of the chromatin on the inside. The chromatin is going to start to coil up to form these X shapes. So this is where these X shapes appear. This is the first time we see these X shapes are in prophase. We have two centrioles that are going to move to the ends of the cell. We can call them like the poles of the cell. You can picture this as like being like the North Pole and the South Pole. And the centrioles are going to start to manufacture these tubes that we call spindle fibers. And so that's kind of going through this little mixture of our chromosomes here in the center. And so these are the key parts of prophase. Metaphase. Metaphase, only important thing here, we'll label it at the end. The spindle fibers will start to go through the centromeres. So yes, there are extra spindle fibers, but they are trying to pierce through the centromere of the chromosome. And like it says here, the spindle fibers move the chromosomes towards the center of the cell. So the reason why the spindle fibers exist are to move and manufacture a nice straight line of chromosomes for our cell division. Now keep in mind, we only have 4 chromosomes in this picture for our cells. Remember, we have 23 pairs of chromosomes. So it would get a lot crazier if we were actually trying to make this accurate to a human cell. That's why we're just saying there are 4 chromosomes in this little picture here. Next, after metaphase, we have anaphase. This is when the spindle fibers separate and rip apart the sister chromatids. So the spindle fibers were piercing through the centromere for a reason, so that they could pull apart the 2 halves of the X's of our chromosomes. And so the spindle fibers literally rip apart the chromosomes, separating them into sister chromatids on either side. So this is still like chromosome 1, chromosome 2, chromosome 3, chromosome 4. It's just we're used to seeing them as like an X, but that's because they're duplicates. They're 2, right? You have to double everything, basically. And so the chromatids are pulled to opposite corners of the cell, and they are literally pulled as far to the side as you can get. So the north and south poles, they're trying to get them as far over as possible, because the last thing you would want is one of these chromosomes just kind of float over, and now all of a sudden you have, an extra chromosome on one side. Last, telophase. So telophase, the spindle fibers are going to start to dissolve. So now notice there are no spindle fibers. Chromatin will start to uncoil, in the chromatid. So now we're getting back to that jumbly, wumbly mess. And the nuclear envelope starts to reform. And that's the end of mitosis. Remember, at this point, they're still attached. You have now 2 nuclei in your cells. In order for that last bit, the cytokinesis part, this will just kind of pinch apart if it's an animal cell until they're separate now. So here is a pretty good representation of the stages of mitosis. So we are going inside of a cell. You can see that wiffle ball looking thing over there. That's the Golgi apparatus. Those are mitochondria. You can see we have vesicles. There you go. There's the nucleus, the wiffle ball thing. So we're going in. We have this mixture of just random chromatin. And now we have organized chromosomes in prophase. Boom. Nuclear envelope goes away. Those are our centrioles. Spindle fibers are starting to get manufactured. The spindle fibers are actually what line them up, so that's a little bit inaccurate because, they don't just do it on their own. That's metaphase, though, where they're all lined up in the middle. And then the spindle fibers will literally rip them apart. Anaphase, you can see they're going all the way to the corners, and now we reestablish our nuclear envelope, and that's telophase. We're now zooming out. We have 2 complete nuclei. And eventually, now we have cytokinesis happening. Now we have 2 identical cells. And that's the end of mitosis. So again, if you have any questions, let me know.